Good day, everyone. I am Charmie Arong, the first reporter of Group 5. Today, I will discuss about the basic process of gene therapy. And the second reporter is Mr. Jonathan. He will be discussing about the two types of gene therapy. And the third reporter is Miss Casey. She will also discuss about the stem cell gene therapy. And the fourth reporter is Miss Labajo. She will discuss about the bioethics of gene therapy, and she will identify relevant global current challenges or issues or problems related to our topic and present solutions on how to address these issues. Lesson 5. The aspects of gene therapy. Lesson objectives. At the end of this lesson, the students should be able to first, describe gene therapy and its various forms, and assess the issue's potential benefits and detriments to the global health. By the way, what is gene therapy? According to my research, gene therapy is the transplantation of normal genes into cells in place of missing or defective ones, in order to correct genetic disorders. It is a medical field which focuses on the genetic modification of cells to produce a therapeutic effect or the treatment of disease by repairing or reconstructing defective genetic material. In simple words, gene therapy involves altering the genes inside your body cells in an effort to treat or stop disease. Genes contain your DNA, the code that controls much of your body's form and function, from making you grow taller to regulating your body systems. And the main purpose of gene therapy is to treat diseases at the genetic level. Medical science has detected many human diseases related to defective genes. These types of diseases are not curable by traditional methods like taking readily available medicines. Gene therapy is a potential method to either treat or cure genetic-related human illnesses. In 2015, a team of researchers at the Harvard Medical School and the Boston Children's Hospital stated that they were able to restore basic hearing in genetically deaf mice using gene therapy. The Boston Children's Hospital research team also reported that they have restored a higher level of hearing down to 25 decibels, which is actually equivalent to a whisper. They use an improved gene therapy vector developed at the Massachusetts eye and ear that was identified as ANC80, which enables the transfer of genes to the inaccessible outer hair cells when introduced into the cochlea. Human gene therapy was actually first realized in 1971 when the first recombinant DNA experiments were planned. It can be simply viewed as insertion foreign DNA into a patient's tissue that hope to successfully eradicate the targeted disease. It was actually inspired by the success of recombinant DNA technology, which occurred over last 20 years. Without a doubt, gene therapy is the most promising yet possibly unfavorable medical field being studied. Before I proceed to the next topic, please let's watch this video.
Let's talk about your genes. These genes. If you had a hole in your favorite pair, you could fix it by patching it up with new fabric. And if you didn't, you might have a little problem. Now, let's talk about your other genes, the DNA that encodes all of the proteins in your body. Unfortunately, mutations in those genes can cause life-threatening diseases, and sometimes there is no drug or surgery available for treatment. Wouldn't the best strategy be to patch up the mutated gene, or at least deliver a correct copy to cells in the body? That is the hope of gene therapy versus, say, drug therapy. Studies in animal models are very promising, but scientists need to be sure of the long-term safety of correcting or delivering genes in humans. It's a problem worth solving because gene therapy could not only cure genetic disease, it could be a real kick in the pants to cancer.
And the next is the basic process of gene therapy. These are the following several approaches to gene therapy. First, replacement of mutated gene that causes disease with a healthy copy of the gene. Second is inactivation of a mutated gene that is functioning improperly. And the last is introducing a new gene into the body to help fight a disease. So in general, a gene cannot be directly inserted into a human gene or cell. A gene is inserted into another gene using a carrier or vector. At present, the most common type of vectors are viruses that have been genetically changed to carry normal human DNA. Viruses have evolved a way of encapsulating and transporting their genes to human cells in pathogenic manner. When we say vector, is to transfer the desired genes into a target cell. Two main classes of vector. These are the viral vector and the non-viral vectors. Viral vectors are the tools commonly used by molecular biologists to deliver genetic material into cells. And the four main types of viral vectors are adeno-associated viral, adenoviral, lentiviral, and retroviral. To fully understand what is vectors and viral vectors and its types, let's watch this video.
What is a viral vector vaccine? A virus is a bundle of genetic code surrounded by a shell of proteins. These proteins are what activate the body's immune system. A vaccine activates your immune system ahead of time, so it's ready to fight off that virus before you get sick. A viral vector vaccine uses a piece of viral genetic code with instructions for your cells to make those proteins that activate the immune system. This genetic code can't just be added to a person's body, it needs a package or container to carry it into a person's cells. In a viral vector vaccine, the package is the shell of a different, harmless virus, called a vector because it carries the genetic code through the body and into a person's cells. All viruses are excellent at carrying genetic material into cells, and even harmless viruses still strongly activate the immune system, so a vaccine of this type may only require one dose. Also, these vaccines cannot cause the disease because they don't contain the whole virus. This type of vector vaccine was first approved for use against Ebola in the summer of 2020, and scientists are still studying its long-term immunity. Years of research on viral vector vaccines prepared scientists to respond quickly to this new outbreak with this new type of vaccine. It holds promise as a fast, effective way to defend against future diseases.
Non-viral vectors, naked DNA, particle base, and chemical base. They are administered by direct administration, plasmid DNA, naked DNA, chemical, and physical. Most of the cardiovascular clinical trials use non-viral vectors as a mode of gene transfer. And that's all about my part, and the next reporter will continue our topic. Thanks for listening.